My name's Mrs. Tyler, and today we're going to do some biology GCSE revision looking at patterns of inheritance. This is for dual and triple award GCSE, and it's for higher and foundation levels. We're going to make something that looks a bit like this. So all you need is a sheet of paper in landscape, and then we're going to break it down into looking at sex inheritance, genetic diagrams or Punnett squares, inherited disorders, and then lastly a quick look at embryonic screening. Right, let's get started. So I'm going to put the title in the middle as usual. Today's title is Inheritance. And I'm going to divide my piece of paper in half down the middle. And then this side needs to be divided into 1/3 and 2/3, so a slightly smaller bit at the top. And this one needs to be 2/3 and 1/3, so a slightly smaller bit at the bottom. So about like that. In this one, we're going to be talking about sex inheritance. So we're going to be thinking about the X and Y chromosomes. So sex inheritance. In this one, we're going to be thinking about genetic diagrams, in other words, Punnett squares. In this one, we're going to be thinking about inherited disorders. And in this one, we're going to be thinking about embryonic screening. All right then, so let's start with sex inheritance. When we're thinking about sex inheritance, in other words, whether an organism is male or female, we're thinking in humans about the 23rd pair of chromosomes. And they are called the sex chromosomes. In females, the shorthand symbol for female is like that, a circle with a little cross on it. There are 2 X chromosomes. In males, however, they have 1 X, which is the same structure as that one, obviously, but the other chromosome is smaller. We call it Y. It's not really a Y shape, it's just shorter. So that is male, that's the symbol for male, a circle with an arrow pointing up. So XX is female, XY is male. When we're trying to work out the chance or the probability of the characteristics an offspring's going to have, or a child in the case of humans, then we use a Punnett square. So this is the diagram that looks a little bit like a noughts and crosses board. At the top, we put 1 set of gametes. Let's put the female ones at the top. So the mother has 2 X's, which means half of her eggs will have this X. Remember that eggs don't have pairs of chromosomes, they only have single chromosomes. So the other half of her eggs will get this X. So 50% of her eggs will be that X, 50% of her eggs will be that X. So that's the female gametes. If we think about the male gametes then, men are XY, and again during meiosis, when those gametes are made and these chromosomes are separated out into single ones, it means half of the sperm will be X, and the other half of the sperm will get this chromosome, the Y chromosome. So that's the male gametes. All we need to do next then is work out the combinations of chromosomes. So if this sperm fertilised this egg, the offspring would have 2 X's, so it would be female. If this sperm fertilises this egg, we'll get 1 X from the sperm, 1 X from the egg, so the offspring will be female. But if this sperm fertilises this egg, it will get one X from the mum, but it will get a Y from the dad, which means that offspring will be male. And if this sperm fertilises this egg, exactly the same again. One X, one Y from the dad. Whenever we draw a Punnett square like this, one of these tables, it's really important that we make sure we label it. So I'm going to label them like this. I'm going to say that there is a 50% chance of it being male, and therefore there's a 50% chance of it being female. If I was doing that as a ratio, because those two numbers are the same, 50 to 50 is equivalent to a 1-to-1 ratio. All right, so equal probability. Remember as well that this is just probability. This does not mean that these people are going to have 4 children and 2 of them will be boys and 2 of them will be girls. It means that every fertilisation, this resets, and it's the chance of a male and female. And I'm sure we can all think of families where they've probably had 3 or 4 or 5 children, all of the same sex, just by chance. So let's take then what we've done in this diagram and put it into looking at other characteristics, not just whether you're going to be male or female. Remember that in genetic diagrams, we are predicting the chance of inheriting the characteristics, not actually getting them. Predicting the chance, or probability of inheriting characteristics. Now this time, instead of thinking about whole chromosomes, we're going to be thinking about types of genes. We're going to be thinking, therefore, about alleles. So our first key word that we need to remember to try to use is allele, which means a different form of the same gene. So alleles can be dominant, which means you only need one copy of them from one parent, or alleles can be recessive, which is where you need 2 copies, one from both parents. And we'll go on to talk about this in a minute. They can be dominant, which means you only need one copy. Or they can be recessive, which means you need 2 copies. All right then. So when we're talking about the combination of alleles, or the combination of genes, we're really talking about what we call the genotype. Genotype, as the name suggests, is thinking about the types of genes. It should be called the allelotype, though, really, because we're thinking about the combination of alleles, the formation of genes. So the combination of alleles. When an allele is dominant, then we give it a capital letter. So somebody might have one dominant allele, and we give it a capital letter B. In this case, let's say we're talking about brown hair. And they might have a recessive one. We give the recessive allele a lowercase letter. So in this case, it's a little b. So that would be for blonde hair. So in this case, the genotype of this individual would be big B, little b. All right. And the genotype is always 2 letters because it's a pair of alleles. Another word we need to know when we're answering these questions is phenotype. And phenotype means the physical characteristics. So for pheno, for physical. So by physical characteristics then, instead of saying big B, the phenotype of this person would be brown hair because they contain the dominant brown allele. So the genotype is the combination of alleles, usually 2 letters, and the phenotype is physically what protein's made, in this case, brown hair. 2 more words we need to make sure we know then. One is homozygous. Homo means the same, which in this case means both alleles are the same. So for example, using this one again, if somebody had the genotype big B, they would be homozygous dominant for brown hair. They've got 2 alleles for brown hair, and they're the same. If somebody was little b, they'd also be homozygous, but this time they'd be homozygous recessive. They've got 2 recessive alleles, so this person would have blonde hair. And the last word we need to know, nearly done, is heterozygous. Hetero means different. So this is when alleles are different. Both alleles are different. So this would be big B, little b, the same as this one up here. So the person would have the phenotype of brown hair, but they'd have 1 dominant and 1 recessive gene. And because they've got that recessive gene, it means that they could have offspring, they could have children with blonde hair because they carry the blonde-haired allele. Right, let's have a look then at a Punnett square of this. Remember, Punnett squares are just predicting the probability. Or chance. So we're going to use this example again. Let's pretend then, who did we do at the top last time? We did the female at the top this time, so we'll do the male at the top this time. Let's pretend that the male has the genotype big B, little b. That means half of his sperm are going to be big B, and the other half of his sperm are going to be little b. Let's put the mum on this side this time. She's also heterozygous. She's got 1 dominant brown-haired gene and 1 blonde-haired gene. She'll have brown hair, but she's carrying the blonde-haired allele. Half of her eggs will therefore have the big B. Half of her eggs will therefore have the recessive little b. Let's turn those into eggs. Let's turn these into sperm. So if this egg is fertilized by this sperm, that child is going to be homozygous dominant. It's got 2 dominant alleles. It will have brown hair. If this sperm fertilizes this egg, the child will be heterozygous. It will have 1 dominant allele, but also 1 recessive one. It will still, however, have brown hair because it's got the dominant allele. The brown hair beats the blonde hair. If this egg is fertilized by this sperm, again, they will get 1 dominant allele from the dad, 1 recessive allele from the mum, so they will be heterozygous, but they will still have brown hair. However, if this egg is fertilized by this sperm, then this offspring will have 2 recessive alleles. They'll be homozygous recessive, which means they will have blonde hair. I need to remember to label that diagram because this is only worth 1 mark in an exam. By labelling it with the probability and the phenotype, that's where I get my other 2 marks. So I need to say it's a 25% chance. And I need to say what the phenotype is, and this child will have blonde hair. Another way I could do it is by putting it in a ratio whereby there's the probability of 3 having brown hair and 1 having blonde hair. So it could be a 3-to-1 ratio of brown to blonde. On to inherited disorders then. And this is basically using the same method as over there, using a Punnett square, but this time we're looking at two major disorders where there's been a change in the sequence of DNA, meaning there's a difference in the phenotype, the physical characteristics. The two we need to know about are cystic fibrosis and polydactyly. Let's start with cystic fibrosis then. Cystic fibrosis is an inherited disorder that makes the mucus thick. Thick mucus. And because it makes mucus thick, when that mucus is in the lungs, it reduces oxygen diffusion. And when it's in the digestive system, it reduces the action of enzymes. So in the small intestine, it reduces the action of enzymes. And the important thing you need to know at GCSE about cystic fibrosis is that it's a recessive allele, which means that the dominant form is healthy, so I'm going to call it F for fibrosis. Capital F for dominant means healthy. And in this case, cystic fibrosis is recessive, so I'm going to use the lowercase f for somebody that suffers from cystic fibrosis. Again, we're going to do a Punnett square. So we're going to draw out our little table. And again, we're going to put the genotypes of the parents. In this case, we're going to say both parents are heterozygous. So they've got one dominant healthy allele and one recessive cystic fibrosis allele. Remember that this means that they are healthy, their phenotype is healthy, but they carry the cystic fibrosis allele. That means half of the eggs in this case, I'm going to do this as the female, the mum, will have the healthy allele, and the other half of the eggs will have the recessive allele. Here's the dad, and again, he is heterozygous, he's a carrier. He's got one dominant healthy allele and one recessive one. And so when we look at the pattern of inheritance, if this sperm fertilizes this egg, the offspring will be homozygous dominant and will be healthy and will only be able to have healthy children. If this sperm fertilizes this egg, the baby will be heterozygous. They'll be healthy, they'll have the healthy phenotype, but they will carry the cystic fibrosis gene, which means that they will be able to have children with cystic fibrosis. If this sperm fertilizes this egg, again, they are heterozygous. Their phenotype will be healthy because they've got the healthy dominant gene, but they're carrying the recessive one. And lastly, if this sperm fertilizes this egg, then the child will be homozygous recessive. They'll have two copies of the recessive allele and they will have cystic fibrosis. So again, this bit's only worth 1 mark. To make sure that I get the rest of my marks, I need to label it. So that's a 25% chance of cystic fibrosis. And again, if I wanted to do it as a ratio, it would be a 3-to-1 ratio of healthy to cystic fibrosis. The other disorder then you need to know about is polydactyly. And polydactyly means having an extra finger. So I'm going to draw a weird hand there with the thumb, and then 1, 2, 3, 4, 5 fingers. Okay, so polydactyly means an extra finger. And polydactyly is a dominant allele, which means you only need 1 copy, remember. So I'm going to use a little code for these alleles. I always use a capital letter for the dominant one, remember. So I'm going to use D for dactyly, because if I use P, a capital P looks the same as a lowercase p, so I'm not going to use P. So a capital D means polydactyly. I have an extra finger, which means little d equals normal. Or this is why you can't say it, 4 fingers and a thumb. Let's have a look then at a Punnett square. Again, we're going to draw out the mum and dad. I'll put the dad at the top this time. And this time, the dad has extra fingers. So he's got the dominant polydactyly gene. He carries the normal gene. Half his sperm will be extra fingers. Half his sperm will be normal amount of fingers. The mum does not have polydactyly. She has 1 thumb and 4 fingers, which means half her eggs will have that allele. The other half will have that allele. As we go through then, looking at the sperm and egg combinations, if that sperm fertilizes that egg, then that child will have an extra finger. If this sperm and this egg are fertilized, then the child will have the normal number of fingers. If this sperm and this egg are fertilized, the child will have extra fingers, will have polydactyly. And if this sperm and this egg are fertilized, the child will have normal number of fingers. So in this case, where only the father had polydactyly, there is a 50% chance of any children he has having extra fingers or polydactyly. And again, if we were doing that in a ratio, it would be 1 to 1 because they're even amounts. The last thing we're going to think about today then is embryonic screening. And this is where you take a single cell from an embryo. Remember, an embryo is just a ball of cells, and you test it to see which alleles it has. Now, this can be done in utero, in other words, in the uterus, so a normal pregnancy, or it can be done during IVF, where an egg is being fertilized outside the body. Let's have a look at how that could work then. We're going to start by drawing out a female reproductive system. So this is going to be the top of the uterus. Here are our 2 fallopian tubes. Here's the bottom of the uterus. And here's the vagina. Remember, the uterus has got thick walls. There's the cervix. There. And this is the uterus wall. At the ends of the fallopian tubes then are the ovaries, and that is where the eggs are released from. So if a woman is having it tested by a normal pregnancy, then a cell is removed from the embryo in the uterus. But if the woman is having IVF, then the first thing that needs to happen is the woman needs to have an egg removed from an ovary. It then needs to be fertilized, and this is done in a tube. So we put the egg in the tube, we put some sperm in, and we let it grow into a ball of cells, an early embryo. So there's our early embryo. And then what they do is they take a single cell out of that and then implant the embryo back into the uterus for it to grow and use the mother's blood supply there. So embryonic screening can be done naturally. So once the baby is growing in the uterus, the woman can have a test where an egg cell is removed, or it can be done via IVF, where the embryo grows or the egg is fertilized, making an embryo in a tube. You then take a cell off there before you implant it. So IVF equals in vitro fertilization. In vitro just means outside of the body. But this can also happen, embryonic screening, during a natural pregnancy. Now, we've got to think about the advantages and disadvantages of parents knowing if their children are going to have an inherited disorder or not. So the advantages of finding out if your child has an inherited disorder is that ultimately it's going to reduce human suffering, because it means that you're not going to have a child that has an inherited disorder. So it's going to reduce human suffering. Ultimately. However, we also need to think about the disadvantages of this. For a start, if you are not having an embryo implanted, so if you have the embryo tested and it tests positive, let's say for cystic fibrosis, and then you don't have that embryo implanted, so you choose a different embryo and that one gets destroyed, then you are implying that cystic fibrosis is undesirable. It also leads to the destruction of embryos. So you're implied that disorders are undesirable. It will lead to the destruction of embryos. And some people say, well, where does it end? So you're saying you don't want a baby with cystic fibrosis. What else are you going to choose? You're going to choose intelligence, height, eye colour. So the designer baby issue is a big one. So where does it end? So some people would say also that it's playing God. Kind of falls into that argument. And that's it. That's everything you need to know about inheritance. So you know your job now is to read, cover, recite. So recall that females are XX, males are XY. Make sure you know these terms here. So read, cover, recite the definition of allele, genotype, phenotype, and homozygous. And then go away and have a go at some exam questions. And I'll see you next time.